Right, morning everyone. I think we'll make a start. So welcome to week 10, so obviously this is the last week. So what we're going to be covering this week is transplant immunology. We'll continue on with that tomorrow as well. So just to run through the learning outcomes for today's lecture. So you'll gain an understanding of the concept of organ transplantation, as well as an understanding of the immunological challenges that we face when we're looking at transplantation. Also, knowledge of the mechanisms of graft acceptance and rejection, and also we'll have a look at bone marrow transplants as well as graft versus host disease, which we kind of touched on a few weeks ago. So I'll start off with the attendance pin whilst I remember. So the pin today is 2125. 2125. So first of all, if we look at transplant immunology, why is transplant immunology so important? Well, we know that in today's medicine, that essentially we have this great need to be able to replace diseased organs, and obviously we've had surgical advances that have enabled us to do so, and that's actually quite a common place, an important part of medical therapy. But we're still faced with this major problem, which is rejection from the host immune system. So if you think of that in terms of an adaptive immune response, and obviously that's quite actually sort of an impediment to transplantation. So why is that? Well, again, I just mentioned it's based on the adaptive immune response. So essentially, we've got this, the immune system recognizing self versus non-self. So therefore, any tissue that's not going to be genetically identical to ourselves is going to be treated as foreign antigen, and it's going to invoke an immune response in the recipient. And that sort of immune response, or rejection, is essentially mediated by a T-cell response to alloantigens. So these are foreign donor antigens, and are usually down to the polymorphic MHC. So remember we talked about MHC being a unique sort of immune fingerprint for each individual. Now obviously the degree of the immune response that's going to be elicited to a transplant will kind of vary depending on the source of the grafted tissue. Essentially, the main goal of transplant immunology is to prevent the recipient's immune response from rejecting that graft or that donor tissue. So we have several different types of transplant. We can have an autograft, so this is from one part of the body to another, so that might be like a skin graft following a burn or something like that. You then have an isograft, so this is a syngenic graft, so this is between two genetically identical individuals, so twins, in the case of humans, or we have an allograft, so this is between two different individuals within the same species, and this is called allogenic. And then of course we have a xenograft, so this is from a different species. So typically for xenograft, we've used pigs in the past, so can anyone think why pigs might represent a good sort of option for doing a xenograft? Any ideas? Sometimes it's just it's more convenience in a way, because they're a similar size, so the organs are appropriately sized, so obviously that's something you want to think about. But also, in terms of their breeding, they reach sexual maturity quite early, and they have large litters, which means obviously in terms of producing viable organs, it's quite an attractive option. And they're routinely used for things such as heart valves, and it's been quite successful in that respect, and obviously a lot of researchers looking into that, and we'll touch on that a little bit more tomorrow as well. When it comes down to transplant rejection, a lot of this was kind of building off the research from these two individuals. So we have these two surgeons, so Thomas Gibson and Peter Medawar, and in the 1940s they really sort of began to understand the underlying issues of rejection. So they were actually sort of looking at skin grafts in pilots that had suffered sort of severe burns during the Second World War, and they realised that this was essentially down to this host versus graft reaction. And they actually published this paper, The Fate of Skin Homografts in Man, and that kind of set sort of the underlying scientific basis for our understanding of transplantation and where we are today. But really, because they worked together, they actually provided the first evidence that allograft transplant rejection was really down to the effects of the immune system. So as I mentioned before, you can think of this in terms of the adaptive immune response, but essentially the same way that an immune response would treat a pathogen. Essentially, that's the same sort of immune response that's being elicited to transplant antigens once we've had transplant. And you can see that on the two slides here. So on the left, we've got normal tissue, and then on the right-hand side, we've got tissue that's undergoing sort of like a rejection, and you can see this massive infiltration of lymphocytes, sort of with the darker staining. So that's kind of giving sort of an impression of what's going on sort of at the tissue level. But really, when it comes to looking at allograft rejection, it helps to have an appreciation of immunological tolerance, but also immunosuppression. Now, this cartoon is just kind of summarizing, obviously, the main steps that you would need for a successful transplantation. But generally, the conditions that you would need you would want the MHC to be as similar as possible between the donor and the recipient. So this is where you have tissue typing and that kind of things. You also want the cell or the tissue, the graft, to be in the best possible condition, so ideally free from disease or damage. And then, of course, you want the correct surgical or transfusion techniques to actually sort of help the engrafting process. Now, when it comes to rejection, we can kind of classify it into two sort of main processes. So the first one, and we actually touched on these a few weeks ago, so hopefully you remember that, but the first one is host versus graft. So this is where we've got the mature T cells in the host that are recognizing the graft tissues as foreign, and then we're getting an immune response mounted against that. And this is also sort of more widely known as graft rejection, and obviously that's what we'll focus on today. And then the second one is graft versus host disease. So this is what you would see after a hematopoietic stem cell transplant. So this is where mature T cells or passenger sort of lymphocytes in the graft are actually detecting the host cell as foreign, and they're sort of then attacking the host or the recipient. So host versus graft and graft versus host are two things that you need to be aware of when it comes to rejection. And so, as I mentioned before, this is all based sort of obviously an immune response, so the adaptive immune response. So graft rejection is antigen specific. It's also escalating, and it involves memory. So the first rejection response would be sort of a slow one where your immune system is getting primed. But then if you encounter tissue from that donor again, you're going to have a second set rejection, which is much faster, and that's obviously mediated by your immunological memory. But rejection can be broken down into sort of four classifications. We have hyperacute, which is very quick, and this is due to sort of pre-existing antibodies. We then have acute, which is about 2 weeks or so. And then we have accelerated acute, which is much faster. So if you encounter tissue from the same donor a second time, obviously you've got that memory, it will be rejected faster. And then you've got chronic, which is much slower and tends to sort of progress over several years and is quite hard to manage. This table just kind of summarizes the four classifications. As I mentioned, hyperacute, it's very quick, minutes to hours, and it results sort of in this immediate graft failure, and it tends to be irreversible and untreatable, so you really need to kind of remove the graft if this happens. And it's kind of down to preformed antibodies, either from a previous transplant or a transfusion, or in some cases, pregnancies. We then have acute and accelerated acute, so this typically will occur within a couple of weeks. And this is due to MHC class 1 and MHC class 2 sort of activating the cell-mediated rejection. So we have this T cell component going on here. This is treatable and is reversible, so it can be managed with immunotherapy. And then we have chronic, so this is what develops sort of slowly over many years. And it's essentially a combination of the humoral response, but also the cell-mediated response. You get this chronic inflammation and tissue remodeling going on, which essentially leads to scar tissue. And this is, unfortunately, untreatable and will sort of result in the overall loss of the... If we look at blood transfusions, so blood transfusions are obviously one of the most simplest and probably one of the most earliest kind of transplants that occurred in humans. But of course, as you all know, we need to sort of perform blood matching. So we really need to sort of be aware of the ABO as well as the rhesus blood group antigens, because if we get that wrong, then we're going to get rapid destruction because we've got antibodies present that will recognize that kind of mismatch. So you can see here that O is the universal donor, and AB is obviously the universal recipient. But when it comes to MHC matching, we don't need to do that for blood transfusions, and does anybody know why we don't need to do that? It's important to blood, to match for the ABO and the rhesus, but why not for MHC? Something we touched on a few weeks ago, but essentially red blood cells don't have MHC. Okay. But because there are only 4 ABO blood types, so we've got O, A, B, and AB, and then 2 rhesus types, essentially it's quite easy to perform blood group matching between the donor and the recipient. So it's actually quite an easy thing to do. Let's have a look now at hyperacute rejection. So this is where we have antibodies that are going to react with the endothelium. So it's really important to not kind of mix up endothelium and epithelium, because sometimes people do that, but just kind of get it straight in your head. So obviously we're dealing with endothelium here. So this can occur following pregnancy, blood transfusions, or sometimes previous transplants. And these can all elicit a hyperacute rejection response. And the immune mechanism that's sort of behind this is that the recipient will have pre-existing antibodies that will recognize that donor graft or transplant. And these alloantibodies can either be against blood group antigens or the polymorphic MHC antigens, as we mentioned earlier. And what this will do is it will cause a really rapid rejection of the graft. So you'll get the antibodies binding to the endothelium, you'll get recruitment of neutrophils and activation of complement. And this all leads to sort of inflammation, and then you start to get this coagulation, and essentially the vasculature starts to gets blocked, and then the graft will die. So if you look at the pathology, you can see sort of you get like thrombosis, you'll get an infiltration of neutrophils, but we'll have little or no lymphocytes present because it's sort of mediated by this preformed antibody. Now, there are ways that we can sort of minimize this, but it's really important to remember that hyperacute rejection, it's irreversible and it will permanently damage that transplanted organ, and so it's really key to perform matching. In that case, we want to be certain that we match the ABO blood type between the donor and the recipient, but we also want to perform cross-matching to determine if there are any antibodies present in the recipient that would react with the donor. And we can do that using the serum panel reactivity antibody test, and we'll talk about that a little bit more tomorrow. But essentially there are a couple of steps that you can perform to actually kind of check that it is a match. But it's really important to do because it's quite a destructive outcome if you get this hyperacute rejection. Now, acute rejection is the most common form. So I'll just bring that up. So, like I said, it's the most common form, and it will occur quite quickly, so within 2 weeks, as we mentioned at the beginning of the lecture. And it's T-cell mediated, and an experiment that actually kind of highlights that is if you take a nude mouse, which is lacking T-cells, it's lacking kind of that immune system. If you perform a transplant, it's not going to be rejected, but then if you perform an adopted transfer of normal T-cells into that nude mouse that has the transplant, you'll kind of restore that rejection capability. So that's kind of proof in a way that you've got this T-cell mediated response for the acute rejection. Now, it can be prevented and controlled by immunosuppressive drugs. And as I mentioned, one of the main targets tends to be the MHC, so the major histocompatibility complex, but also there's an involvement for minor histocompatibility. We'll talk about that in a few slides' time. But there's 2 mechanisms by which the recipient can actually sort of detect the donor antigen. This is via direct presentation or indirect presentation. So we'll look at that slides' time. I just kind of want to have a look at the role of the major histocompatibility complex. Hopefully you remember that the MHC, it's also known as the HLA in humans, so the human leukocyte antigen. And it's really important because, if you remember, the MHC is kind of essentially what gives us the unique immune fingerprinting. So it's like a unique immune barcode, so it's specific to each individual. And that's because it's not only polygenic, but it's polyallelic and it's polymorphic. So if you look at this slide, you can see the sort of the structure of the MHC genes here. And this is kind of highlighting that it's polygenic. So you can see that for MHC class 1, we've got HLA-A, HLA-B, and HLA-C, and a similar kind of structure for MHC class 2. And then it's also polyallelic, so this means that it's got sort of multiple genes at each locus. So for instance, at HLA-B, we also have HLA-B8, B15, and B27, for example. And then it's polymorphic. So what I mean here is that within humans, sort of within a species, you have kind of variation of the genes. So you're getting sort of variation at the amino acid level. And we can see that here if we're looking for sort of MHC class 1 and MHC class 2. So this is just showing sort of variations that are highlighted in red. And you can see that these kind of allelic variations are actually occurring within this sort of peptide binding region. So if you look here, this is kind of like a peptide binding groove. But you can see all these variations are occurring kind of within the N-terminus, within this binding groove. And humans will actually have sort of over 1,000 different alleles for MHC class 1 and MHC class 2. So that's kind of, it gives us that kind of uniqueness that we need for our MHC, and it's really important. But in terms of transplants, obviously that presents a bit of an obstacle because it's obviously going to be different between each individual. If we look at sort of the polymorphism between the different sort of transplant antigens, as we mentioned, we have the ABO blood group, and the polymorphism here is limited because we just have to deal with the ABO system. So these are obviously down to the carbohydrates that we have on the red blood cell surface. And then as we just discussed, we have the MHC, so the major histocompatibility, or the HLA, and the polymorphism here is very high. This is really kind of the main transplantation antigen that we need to be aware of. And then we have the minor histocompatibility. So these tend to be sort of like the non-ABO blood group. They tend to be sort of associated with the sex chromosome antigens, and they will elicit a much weaker response and a kind of late onset response, and we'll discuss that later on. And then, if we compare that to xenoantigens from a different species, obviously these have a very, very high polymorphism because they're from a different species, and that would elicit a very sort of aggressive and rapid rejection. So I just wanted to put this up because it's sort of an experimental model, and it shows sort of how MHC can be a very potent trigger for acute rejection. So this was kind of derived from mouse experiments, and it shows that even if we have a match for MHC, it doesn't necessarily ensure the graft's survival, and that's down to the minor histocompatibility antigens. So here we have two genetically identical mice, and we perform a syngenic transplant. Obviously that graft is going to be accepted because they're genetically identical. If we perform an allograft, so we've got two different individuals here, they've got different MHC. What we'll have here is we'll get an acute rejection, so within about 2 weeks, and the transplant will eventually be destroyed. But if we match the MHC, so this time we've got an individual mouse that's matched for MHC-A, the graft will survive longer, but we'll still get rejection, and this is down to the minor histocompatibility antigens. So we need to factor them in as well, but the graft will be rejected, albeit a lot more slowly. And kind of continuing on with these experiments, this just kind of helps to reinforce the fact that graft rejection is involving T cells, so we've got this T cell mediated response in rejection. So over here on the left we have that same sort of diagram that I showed you on the other side. So we've got basically a syngenic graft between identical individuals, and we get the graft being fully accepted and 100% tolerance and no rejection. In B, we've got an allograft, so we've got genetically distinct individuals. This is where we're going to get acute rejection. So you see after about 2 weeks, we start to get rejection of the graft. Now, if we take an individual that has already had a graft from that same donor and we give them a second graft, this time we'll get rejection, but we'll see that it's much more accelerated. So, we can see that here, it's rejected in about half the time. So this is obviously showing you that we've got a memory response going on here. But what we do is if we take T cells from an individual that has had a graft and we put it into a recipient that is sort of having a graft from the same donor but for the first time, we will essentially elicit a sort of second set rejection response. So that's kind of proving that those T cells are important, not only in memory, but it's also important in kind of mediating that anti-graft response. Okay? So it's just showing the link between the adaptive immune response and graft rejection and the link to T cells. And then looking sort of at the minor histocompatibility, so we talked about that, and this is the same sort of diagram, but essentially here we have a skin graft between two individuals in which they have the same major histocompatibility, so they're identical for the major histocompatibility, but we still get this slow rejection, and this is due to the minor histocompatibility proteins. Now, the minor histocompatibility proteins, they're also polymorphic within the population, and you can see that on the diagram here. So this is sort of a self-protein here on the donor and on the recipient, but you can see that there's subtle differences. So there's, you know, it's the protein, but it has that kind of polymorphism within the population. So it's actually distinct between the two individuals. And what happens is these would be sort of presented on MHC class 1, and that's how it would sort of alert the immune system. I mentioned before, they tend to be non-ABO blood group antigens, but they also tend to be antigens that are associated with sex chromosomes. So obviously, you know, if you think about that, you know, between the sexes, there's going to be a difference there that proteins that the one sex may not have seen. But when it comes to transplantation, there's pretty much a virtual certainty that there will be a mismatch of the minor histocompatibility between individuals. And that's really why we need to use immunosuppressive drugs, even if we have like good matching going on. So as I mentioned here, a successful transplant will involve you know, the use of immunosuppressive drugs to maintain that transplant. I want to have a quick look at direct and indirect allorecognition. But before that, I just wanted to put up this sort of statement that's saying that, you know, if you have a naive alloreactive T cell that needs to become activated and acquire effector activities that would be responsible for mediating graft rejection, that T cell must sort of encounter an antigen-presenting cell as well as co-stimulation. You know, so antigen-presenting cell must also have this allergenic MHC. So that's kind of just showing that you need that adaptive immune response for the rejection. Now this slide is just summarizing the events that go on in direct recognition. So what we have here is we have a donor dendritic cell. So this will be a donor dendritic cell within the graft, so you can see that here. And essentially that donor dendritic cell will be expressing donor MHC, but it will also be expressing donor peptides within the context of that. This donor dendritic cell will then migrate to the lymphoid or the secondary lymph organs, and it will encounter alloreactive T cells, so CD4 and CD8 T cells, from the recipient, and these will become activated by this direct allorecognition. Then what we have is we have the effector cells then migrating back into the graft and then mediating this sort of immune response and essentially attacking the graft cells that are displaying that donor peptide, result in graft rejection and then graft. So that's direct recognition. Now if we turn our focus to sort of indirect recognition, so in this instance we actually have a recipient dendritic cell. So the recipient dendritic cell will migrate into the graft or the transplant, and it will take up allergenic proteins. So these can be minor histocompatibility proteins, which are highlighted in green, but it can also be due to MHC class 1 and class 2, which is allogenic in itself. And these can be released from donor cells by exosomes, and they'll be taken up by that dendritic cell. They'll be processed and then presented on sort of the self MHC class, class 2, to the recipient's CD4 and CD8 cells. And that will sort of mediate immune response within the graft as well. So it's important to just kind of realize the differences between the direct and the indirect recognition of antigens within this sort of rejection response. then this is just kind of summarizing the two. But essentially, with direct allorecognition, it's involving a donor antigen-presenting cell. It can be any peptide, and this is being presented to the recipient's T cell. But then with the indirect allorecognition, we've actually got the recipient's antigen-presenting cell, and the peptide is either donor peptide or it's derived from the donor MHC itself that has been processed and then presented. Now, I just also wanted to bring up pregnancy, because this is kind of a good example of tolerance and the tolerance that is required to provide that protection. But essentially, if you think about pregnancy, the fetus is actually an allograft, and it's not rejected, so you kind of why. But if you think about it, it's an allograft because it's got HLA from both the mother and the father. Okay? Now, even though that fetus is expressing sort of not only the maternal but the paternal MHC class 1 and class 2, it's not provoking a full-on immune system. This is largely down to the placenta. So the placenta is obviously, it's derived from the fetus, and what it's actually doing is it's sort of sequestering the fetus from the mother's T cells and the immune system. And the actual outer layer of the placenta, which is known as the trophoblast, doesn't express MHC class 2, and it's got low MHC class 1. Now, you might think back to a few weeks that, cells that have low MHC class 1 can be a target for natural killer cell, for NK cell sort of killing. But the placenta is able to avoid that, so it's got mechanisms to sort of suppress that natural killer cell targeting as well. We also have the production of IDO. So remember from a few weeks ago, we talked about IDO sort of, and its involvement in a repressive sort of microenvironment, and that helps to reduce sort of the T cell response. And there is actually sort of an experiment that's been performed in mice where if you inhibit IDO in pregnant mice, that this will cause a really rapid rejection of the fetus, and that sort of helps to kind of reinforce that kind of theory. But also you have sort of some other regulatory immune mechanisms. So you have your Tregs, also you have TGF-beta and IL-10 sort of helping to maintain that sort of immunosuppressive environment and make sure that the fetus is essentially protected the maternal immune system. So what's the thinking behind T cells being involved in acute rejection? So this is a slide that's kind of summarizing what we talked about before, but really it's an experimental demonstration that T cells are sort of transferring that or being responsible for allograft rejection. So here we have a mouse receives a transplant and we get our first set rejection. If we then perform a second skin graft from the same donor, we'll get a second set rejection, which is much faster. Okay, so we've already talked about that. Now, if we take splenic T cells and we do an adoptive transfer between a mouse that has had a first set rejection and we put it into an individual mouse that is having a transplant for the first time, we will actually end up with that second set rejection. So that's kind of what we talked about before, but that's really showing the involvement for T cells in this acute rejection. But what is the relationship between the CD4 and CD8 T cells? Is there sort of an importance there? So that's what they've done in this experiment here. So they've used monoclonal antibodies to deplete either CD4 or CD8 T cell populations within a mouse that has had a transplant. So the control is shown in black, and then we've got sort of anti-CD8 depletion shown in red, and then obviously anti-CD4 in blue. So you can see that if we deplete the CD8 cells, we're not getting much difference from the control. If we deplete the anti-CD4, we get sort of a longer survival of the graft. But if we deplete CD4 and CD8, we get an even longer sort of survival. So what it's showing is that the CD4 T cells seem to be more important than the CD8 T cells. But together, they're sort of mediating like a really strong graft rejection. So, hopefully you can see that here. But it kind of ties into what we already know about the immune system, is that CD8 T cells do play a role in graft rejection, but they need that kind of CD4 help. So I think back to your T helper cell, providing that help to the CD8 cells. That's just kind of an experiment that kind of prove the role of the cells in rejection. As we mentioned before, obviously, graft rejection involves sort of effector T cells migrating back into the graft and then kind of mediating that tissue destruction. As we said before, there's obviously an important role for CD4 positive T cells, and we can have those sort of with a Th1 response. So this is important in recruitment of macrophages, and we're getting activation of macrophages, and we're actually getting sort of a delayed type hypersensitivity response that's kind of, sort of eliciting that injury within the graft. There's also sort of recently a role has been shown for Th17 in graft rejection, and then as we saw on the previous slide, there is a role for CD8 T cells as well, because they can actually sort of provide that direct lysis of the endothelium within the graft. But all of this is coordinated by cytokines, and so that's kind of important to remember. So this is just a little bit of a summary. So it's showing that after 24 to 48 hours, we're getting induction of the immune response. So we've had our transplant. In this case, we're looking at sort of like a direct sort of recognition. So we've got the dendritic cell from donor. So it's moving into the lymph node. We're getting sort of activation of the CD4 and the CD8 T cells. We're getting proliferation of these. And this is kind of sort of helping to direct this B cell antibody response and the production of antibodies, activating macrophages, as well as getting this delayed type hypersensitivity response, but then also sort of activation of cytotoxic T cells. But essentially, all of this is going to mediate, the ultimate sort of acute rejection of that graft. And this is just another summary slide. Just showing kind of the different effects. So again, obviously we're getting sort of antibody release, and we're getting antibody-dependent cell-mediated cytotoxicity, so we're getting recruitment of macrophages and natural killer cells. We're also getting a direct action of complement on the graft as well, and then the involvement for cytotoxic T cells as well. But essentially, all of these mechanisms, kind of really having a very severe effect on the architecture of the graft, and will ultimately get sort of inflammation and tissue remodeling, and ultimately. So we have a look now at the cytokines. So we mentioned that really the cytokines are quite important in coordinating the rejection of the graft as well. So we have the classic sort of Th1 cytokines. So we've got IL-2 and interferon gamma. So as you know, that these are going to promote and drive that T cell proliferation. It's also going to help that delayed type hypersensitivity response. And also we're going to get complement activation as well. And interferon gamma and TNF alpha, they also sort of encourage the MHC expression. Now, obviously, if you think in a normal immune response, that's going to be particularly good if you have a viral infection. But these, it's not appropriate if we're having a transplant, but obviously the immune system was not geared to deal with transplants. It's not a normal situation. So, that's things we need to bear in mind. We also have a role for Th2 cytokines. So we've got IL-4, IL-5, and IL-13. So these are helping to drive that B cell sort of activation and expansion. And then, as we mentioned, there's sort of been a recent sort of implication for Th17 cytokines. We also have an antibody-mediated rejection. So this can kind of occur within the early stages of sort of like the graft rejection, but it can actually occur pretty much at any stage. But it sort of helps, the antibody sort of is aiding in that kind of complement-mediated sort of destruction of the. If we look now at chronic rejection, so there's sort of multiple mechanisms that are going on here. But really, it's kind of important to realize that when it comes to chronic rejection, it's very acute sort of subclinical rejection events, and these are constantly kind of recurring. So they're not very easy to detect, but it's just kind of these really sort of reoccurring sort of acute events that just keep happening. And they can be antibody mediated, but they can also be T-cell mediated, or both of them. But essentially, what's happening is we're getting this sort of interaction of antibodies that are recognizing anti-HLA class 1. So you can see we've got the antibodies binding to the endothelium of the blood vessels, and then we're getting recruitment of macrophages and immune cells. And essentially we're getting this inflammation. Okay? So we're getting this chronic inflammation and tissue remodeling, and eventually that will sort of lead to this chronic rejection. But it can occur months or many years after the sort of acute rejection events. So there are obviously quite a few challenges when it comes to chronic rejection, and one of them is this chronic allograft vasculopathy, and this is really kind of a prominent feature of sort of like of graft rejection, especially in heart and kidney transplants. There's several challenges with this. It's difficult to detect, so remember I said it's a lot of kind of recurring subacute or subclinical, acute rejection events, so it's difficult to detect. We're also getting sort of multiple responses being involved, so we're getting the humoral but also the cell-mediated responses. And immunosuppressive drugs will actually be of little help in this instance, but also bear in mind that immunosuppressive drugs themselves can have effect on vasculature, especially sort of cyclosporin A can sort of have that kind of injury on the vascular system itself. And unfortunately, most transplants will experience chronic allograft dysfunction ultimately. So they're the challenges that we're kind of left when it comes to chronic rejection. But chronic rejection, it kind of is dependent on this indirect allorecognition. So this is, as we saw before on the previous slide, we've got the recipient dendritic cell, and it's basically processing and displaying self-MHC from the recipient, or from the donor, on recipient MHC, and it's kind of eliciting that immune response. But it can sort of show itself or manifest itself differently depending on the organ that's being transplanted. So, for instance, in liver transplants, we're getting loss of bile ducts, whereas in lung transplants, we may well get scar tissue. Particularly true in the bronchioles. But there are also other causes that we need to be aware of. So, if you think in terms of immunosuppression, obviously you're suppressing the immune system, and that may allow latent viral infections to actually sort of reoccur and reactivate, and that can have an effect as well. So that's something we need to be aware of. But then also, in some instances, the actual disease that an individual had that destroyed the original organ may well reoccur, and then that can destroy the transplanted organ as well. As we mentioned, chronic rejection, it's irreversible, and it tends to be progressive, and it will ultimately lead to total failure of that transplanted. I want to touch on now on blood transfusion, and an example. This is obviously we've got an example of graft versus host in blood transfusion. So just to refresh your memory, graft versus host is where we're getting the T cells from the donor actually attacking the tissues of the recipient because they're recognizing the host cells as foreign. This can actually occur in between 50 to 70% of bone marrow transplants, so it's really quite high. And it's driven by this HLA or MHC disparity. But despite that, from a hematopoietic stem cell, you only need 10% of that donor bone marrow in order to actually, get a complete restoration of the recipient. They're also beneficial because there's no sort of direct surgery involved, but also hematopoietic cells can actually be banked, so they can be stored. So there's obviously a lot of potential benefits that kind of outweigh the fact that graft versus host is actually, quite a significant outcome that needs to be dealt with. So there's some challenges, but the benefits do outweigh. Now we talked about this before, but in graft versus host, what's happening is we're getting the donor T cells, so the CD4, the CD8 T cells. That are alloreactive to the recipient's MHC, and they're becoming activated by encountering dendritic cells in the lymph nodes or the lymphoid tissues. These T cells are then moving back into circulation, and they're actually attacking the recipient tissues. Now, they tend to do quite a bit of damage on the sort of epithelial cells, so particularly the skin, intestines, also the GI tract. These tend to be organs that may well have been damaged quite significantly by the chemotherapy or radiation conditioning that was required for that stem cell transplant to sort of engraft to make space for. Now, graft versus host disease, it's particularly aggressive when we've got a mismatch between MHC class 1 or class 2. This is why immunosuppression really must be used when we're doing hematopoietic stem cell. Now, the graft versus host disease, this is just kind of displaying the symptoms. So as I mentioned, it can potentially attack every tissue within the body, but it potentially, principally affects the skin, the liver, and the GI tract for the reasons that I mentioned on the previous slide. It can be fatal, and it is a type 4 hypersensitivity reaction. And then we have 4 different grades when it comes to the diagnosis of that, and they're grade 1 through to 4. Listed the sort of diagnoses here. But we can treat the acute graft versus host disease. So we can help to sort of suppress that immune response, that immunosuppression, and try and reduce the number of T cells. So that's where corticosteroids are particularly important in kind of helping with that. And when it comes to chronic graft versus host disease, we can have some options here. So we can use steroids, cyclosporin and rituximab, again, to kind of help just suppress that immune response. But it's something that will need to be done if a patient is having a hematopoietic stem cell transplant. Be kind of wary of these potential outcomes. So that's it for today, and then tomorrow we'll sort of focus on more sort of the options of what we can do when it comes to transplant immunology, and then this is just some further reading from the books, and there's also a little article there if you're interested in reading anything further. But other than that, I'll see you guys tomorrow morning.